Hello to our friends joining us via recording. We are wrapping up our discussion of lesson number 10 about the way that neurons work. To help us review that, we worked in groups to talk about the locations where we have various kinds of channels on our neuron. Now, when we talk about a neuron, for my friends here in the chat, when we're talking about the part of a neuron called its dendrites, what's the function of the dendrites on a neuron? What do those dendrites do? Yeah, so dendrites are the part of the neuron, like we're saying here in the chat, that receive the messages. So when we talk about neurons, when they're talking to each other, dendrites are where a neuron first receives a message, and they'll process that message to figure out whether or not they're supposed to share it with their neighbor. If they decide they're supposed to share it with their neighbor, they will share it at the axon terminal. When I'm talking about the messages that neurons receive, what's the special name of those messages? How does a neuron receive a message? It's not a text message. Yeah, so what a neuron's going to receive, the actual message itself that goes from one neuron to another, is called a neurotransmitter. A neurotransmitter. Neurotransmitters are the names of the chemicals that are the messages that a neuron receives. So when we're talking about the part of the neuron that receives messages, the dendrites, they're going to have the kinds of ion channels on them that are opened by neurotransmitters or that are opened by chemicals. So when we look at our list here of all the various types of ion channels that we find on a neuron, all of my ones that are chemically gated, so all of my CGs, are going to be found at the dendrites. So chemically gated chloride channels, chemically gated potassium channels, chemically gated sodium channels. Because this is the part of the neuron that's receiving chemicals, I'm going to put the channels there that open with chemicals. Now, before we go from the dendrites to the part of the neuron called the axon, there is one part that's in between, or maybe when we go between the cell body of a neuron that has all of its dendrites and the axon of that neuron. What's that place in between? Here, let's do a sketch. Maybe that'll help us to identify it. Here's my lovely neuron, right? I hope you all have had a chance to practice drawing your neurons too. Yeah, so the part we're talking about here, and we are identifying it in the chat, is this part here called the Axon Hillock. I didn't include that on my list. I probably could have or should have included that on my list. Because when we talk about the axon hillock, this is the part of the neuron that does that thing we were talking about that we want to discuss more, the process of summation. Remember that summation is when I add together all of the different messages that I'm receiving to figure out whether or not I should send a message down my axon. Summation is the process that helps me get to threshold. Can you remind me in the chat, what's the number on threshold? What's threshold for a neuron? Here, we'll draw our little graph while we're at it. Yeah, so my value on what's called threshold on a neuron, that's negative 55, negative 55. So here's my little threshold line right here. You told me accurately that threshold is negative 55 millivolts. Negative 55 millivolts is a charge, or it's a voltage, where I start opening up voltage-gated channels. Voltage-gated channels are what's going to help me to get a message all the way down my axon. Remember that the function of the axon is to send messages away from the cell body. So all along the axon here, we have voltage-gated channels. Now, if you look at my list here, there are 3 kinds of voltage-gated channels. One of these 3 kinds is not found on the axon. Which of these 3 kinds of voltage-gated channels is not on the axon? Yeah, so the one that is not on the axon are these ones right here. There are no voltage-gated calcium channels on the axon. The only place on a neuron that has voltage-gated calcium channels are the axon terminals. The axon terminals. These are going to have voltage-gated calcium channels. I know this is a question on our next slide for our next activity, but what's the function of voltage-gated calcium channels? Why do I have those channels down here? Voltage-gated calcium channels. Yeah, so voltage-gated calcium channels, their job is to bring calcium into the cell. When calcium comes into the cell, my neurotransmitters that are hiding down here, they're waiting to be released. When calcium comes in, the neurotransmitters leave. Calcium and neurotransmitters can't be together. They don't mix. So the neurotransmitters spit out at the axon terminals when calcium comes in. So calcium comes in down here at the axon terminals because my action potential, my change in charge, we got all the way up here to positive 30 when we're down at the axon terminal. That's going to lead these channels to open up, bring in calcium, so that we spit out a neurotransmitter. So here's a wink, wink, nudge, nudge when we're working on the homework assignment. Don't tell me that the axons have voltage-gated calcium channels, because they don't. And we can remember that because the job of voltage-gated calcium channels is to spit out neurotransmitters. And the only place I spit out neurotransmitters is at the axon terminal. So I've only got these guys at the axon terminal because this is how I get my neurotransmitters to spit out. Now, to get an action potential all the way down here, to get my membrane charge to go up to positive 30 at the axon terminal, I have to use my 2 other kinds of voltage-gated channels. So when a neuron determines that we've hit threshold and we want to send a message to our neighbor, I need to send that positive signal all the way down my axon. The way I'm going to do that is using voltage-gated potassium channels and voltage-gated sodium channels. Here's a visual I want you to consider when you're trying to understand the way that neurons work. This little graph over here, what I did with my membrane potential, pretend that each place along this axon is doing the exact same thing. So all of my dendrites, they're receiving messages. We're putting those messages in the blender. We're doing summation right here. If this part of my neuron, if the Axon Hillock gets to negative 55, we get to threshold, that's going to cause my voltage-gated sodium channels right here to open up. And when these channels open up, they're going to make this next part of the membrane get to negative 55, which means that the voltage-gated sodium channels here are going to want to open up. Well, when they bring in some sodium, that makes this part of the membrane get to negative 55, and that's going to bring it all the way up. So this part of the membrane is going to depolarize. Here's the visual version of what I was just saying. Every part of my membrane, pardon my terrible drawings here, every part of my membrane has to do this change in charge process. It starts at resting, it's going to get all the way positive, and then we're going to shoot back down negative. First, this part depolarizes, which causes this part to depolarize, which causes this part to depolarize. When we get all the way to the axon terminal, that's the last place where we're going to depolarize the membrane. And since we're at the axon terminal up here, we'll open up voltage-gated calcium channels. So I use my voltage-gated potassium and sodium channels to send a message all the way down the axon. When I get to the axon terminals, at the axon terminal, I'm still going to use a voltage-gated sodium channel to depolarize my membrane. But this time, since I'm at the axon terminal, I'm also going to use those voltage-gated calcium channels to spit out a neurotransmitter. And once I've spit out the neurotransmitter, I need to go back down to normal. So I'm going to use my voltage-gated potassium channels to get back to normal. There was a good question in the chat about the kinds of channels that would be here at the Axon Hillock. The Axon Hillock does summation, but it does not receive messages. If I'm not receiving messages here, would I or wouldn't I want to have chemically gated channels? If I don't receive messages, are chemically gated channels helpful? Yeah, Nicole's saying I would not want to have chemically gated channels because there's no chemicals coming here. This is the first place. The Axon Hillock is the first place that I have voltage-gated potassium and voltage-gated sodium channels. So the Axon Hillock is the first place where it starts to matter if I get to threshold. All that my dendrites have on them are these chemically gated channels. So they're bringing in some chloride, or they're bringing in some sodium, or they're bringing in some potassium, or they're letting out some potassium. All of the ion stuff that goes on here from the dendrites in the cell body, I mix all of that together to see what happens to my membrane charge here in the Axon Hillock. If I get to -55, voltage-gated channels here will open up, and then I'll send that message down this direction using those voltage-gated channels. So if we wanted to add one for ourselves here, the Axon Hillock, the types of channels that we have there are voltage-gated sodium channels to depolarize the membrane, and voltage-gated potassium channels to repolarize the membrane. Remember, the only place we have voltage-gated calcium channels is at the axon terminal, at the end of the road. Because the one goal in life of bringing calcium into my neuron is to get my neurotransmitters to spit out. That's the chemistry of getting us to spit out neurotransmitters. If I bring in calcium, neurotransmitters leave. They can't stay. They got to go. Yeah, so to Jacqueline's question, she asked why exactly we release calcium. The only reason that we do anything with calcium is to get those neurotransmitters to spit out. It's kind of like how water and oil, they can't be together, right? Hydrophobic and hydrophilic, they can't be together. Calcium and neurotransmitters can't be together either. If calcium comes into the axon terminal, neurotransmitters have to leave. But that's a good thing, because then I'm sending a message out to my neighboring cell. I'm talking to them. Yeah, so the goal of releasing a neurotransmitter, that's the way I talk to my neighbors. Absolutely. So I got to spit out neurotransmitters to be able to talk to my neuron. And yeah, Hannah is right in answering Nicole's question there. The only place that I have voltage-gated channels, the synaptic end bulb, which is the same thing as the axon terminal. Here, let me add that terminology. Synaptic end bulb. Gotta love how anatomy has multiple names, right, for the same thing. Synaptic end bulb. That's the same thing as the axon terminal. Any other questions before we move on from here? We've had some good questions. Okay, well, I'm not getting anything in the chat, so I'll go ahead and bounce to our next one. We've already answered some of these questions, especially this one right here. Which of my types of channels was the one that makes me spit out neurotransmitters? Who did we say does neurotransmitters again? Yeah, neurotransmitters, it's all about the calcium channels, the voltage-gated calcium channels. So that's your purpose when you're memorizing voltage-gated calcium channels. Their purpose is to help us spit out neurotransmitters. Hey, here's a wink, wink, nudge, nudge. On the homework assignment, you will also see listed as your answer choices the option of chemically gated calcium channels. Chemically gated calcium channels. Have we talked about chemically gated calcium channels? Have we mentioned them before I just typed it on here? Yeah, we're hesitant, right? But we have not talked about them because, correct, like Jacqueline said, it's not actually a thing. Think about the function of calcium is to get us to spit out neurotransmitters. I only find chemically gated channels at the dendrites. Chemically gated channels at the dendrites, we would never spit out neurotransmitters at the dendrites. The only place I spit out neurotransmitters is at the synaptic end bulb or at the axon terminal. That's the one I like better. So chemically gated calcium channels, not a thing. Voltage-gated calcium channels, a thing. They help us spit out neurotransmitters. They don't mess with membrane charge. It's just those neurotransmitters. I only find them at the axon terminal. Yeah, so Nicole says she thinks I asked about this on Wednesday. I probably did because I always get student questions on the homework assignment. They're like, but chemically gated. Well, yes, chemically gated, but like Christina said, we don't want neurotransmitters being spit out at the dendrites. That's not where we're talking. So chemically gated channels always found at the dendrites, but I don't have chemically gated calcium channels. Things that don't exist, things that would be problematic for me if they existed too, because I'm not trying to spit out neurotransmitters up there. All right, let's go up here. Are types of channels that make things more negative. Now, there are a couple of places where my membrane charge gets more negative. One of the places where my membrane charge gets more negative is at those dendrites. When I'm at the dendrites, which kinds of channels make my membrane charge more negative? Yeah, so Jacqueline posed a good question in the chat, and that's why it was kind of tricky. Jacqueline asked, are we talking EPSPs or IPSPs? We're talking about the whole action potential. We're talking about both. Yeah, we're actually talking about both. So yeah, Nicole put a great answer there. When we're talking about what makes things more negative, we're talking about more negative. Anything that has to do with potassium or anything that has to do with chloride is going to lead my membrane charge to get more negative. So when I'm at my dendrites, when I'm talking about the place that receives messages from a neighbor, what's going to make my membrane charge more negative are those chemically gated chloride channels or the chemically gated potassium channels. Now, when I'm working my way down the axon, when I'm trying to repolarize my membrane, say it's at positive 30, and I got to get it back down, I'm not going to use chemically gated channels at that point. At that point, I have to use voltage gated channels. So the other type of channel that's going to make my membrane more negative are those voltage gated potassium channels. So voltage gated ones on the axon, they're on at the axon terminal. Oops. When we're talking about the chemically gated ones, remember Chemically gated ones only found at the dendrites. Only found at the dendrites. Okay, let's do the same thing, except over here with positive. When we're talking positive here, which kind of ion helps me become more positive? What makes me more positive? Yeah, so at the dendrites, what's going to make me more positive? Those chemically gated sodium channels, because sodium can rush into my dendrites, make me more positive. Once I have enough positives when I'm doing summation, now I'm going to activate those voltage-gated sodium channels. The voltage-gated sodium channels are going to allow me to do something with that. So the reason these questions were tricky was to get you thinking big picture, which it sounds like we kind of did for the most part. When we talk about making the membrane more positive, whether we're talking about making a small change, or making an EPSP, or whether we're talking about completely depolarizing the membrane, it doesn't matter whether it's a big change or a small change. If it's a positive change, it's always going to be sodium. Sodium is always what makes us have a positive change. If we're talking about making the membrane charge more negative, whether we're making an IPSP, where the membrane charge is more negative, or whether we're repolarizing the membrane to get it back to normal, I'm always going to use potassium to be more negative. Or if we're at the dendrites, I could also use chloride. That would be the other option to make it more negative there. Let me pause for a moment. Do we have any questions about this? Yeah, go for it, Hannah.
Sorry if you already said why you did this, but just to clarify, so for these channels that make the membrane charge more negative, when you circle the Vg potassium channels, you put axon and axon terminal. Did we talk about the calcium channels being the only one that's being on the axon terminal, or why did you put that there?
No, it's a good question. We have actually 3 kinds of channels at the axon terminal. The first 2 types of channels that we have at the axon terminal are the same ones that are on the axon. So the sodium channels and the potassium channels. Here, what I should have done, let me change my color down here. My last one too, I should have circled this one in blue since we're at the axon terminal. For us to be able to get a voltage-gated calcium channel to open, I first have to bring in sodium. So the job of a voltage-gated sodium channel is to get us all the way up to positive 30. And I have to do that at the axon terminal, just like everywhere else. When I'm at the axon terminal, in addition to opening a potassium channel to repolarize the axon terminal, I'm also going to open up a calcium channel to help me spit out the neurotransmitters. So what's unique about the axon terminal, as opposed to the rest of the axon, is in addition to having the sodium channels and the potassium channels that make the charge go up and then go back down, the axon terminal also has these voltage-gated calcium channels. The only place I find them is there at the end. But to get them to open, I'm also going to have our friends here, the voltage-gated sodium channel and the voltage-gated potassium channel. Did that answer your question, or did I dance around your question? Okay, perfect. All right, so the one last thing we did in groups that it ate my slide for was we reminded ourselves of a little bit of the terminology that we ended with last time. So EPSPs and IPSPs. EPSPs, when you're studying, I think you should just memorize their acronym. If you want to memorize the words, feel free. Excitatory postsynaptic potential is an EPSP. An IPSP is an inhibitory postsynaptic potential. Let's start with the basic questions, the easiest questions about these guys. An EPSP, excitatory postsynaptic potential, or an IPSP, an inhibitory postsynaptic potential, who's going to make the membrane charge get more positive? Who makes membrane charge go up? Yeah, so excitatory postsynaptic potentials, they make that neuron get excited. An excited neuron is a neuron that talks to its neighbors. So EPSPs are when I change the charge on my plasma membrane in a way that makes me more positive. It's going to get me closer to threshold, because if I get to threshold, I'll be able to open those voltage-gated channels that I need to get all the way to positive 30. So an EPSP, an excitatory postsynaptic potential, makes it more likely that I'm going to get to threshold. That means that an IPSP, an inhibitory postsynaptic potential, that's a change that happens in my membrane charge where I become more negative. I'm inhibiting my neuron's ability to get to threshold, because if I can't get to threshold, I'm definitely not going to be able to talk to my neighbor. So an inhibitory postsynaptic potential makes me more negative, makes me less likely to be able to talk to my neighbor. Now, what our questions were supposed to tell us, that the slide 8 here, was those different kinds of chemically gated channels that we talked about on the previous slide. So here's back to those questions that we first answered. When we were talking about our types of chemically gated channels, which types can be used to make the membrane charge more positive? Which ion makes my membrane charge more positive? Yeah, sodium makes my membrane charge more positive. So when we are talking about the type of channels that helps us to make EPSPs, excitatory postsynaptic potentials, this is chemically gated sodium channels. Chemically gated sodium channels. Here's a note that I want you to write down for yourself, just to make sure we keep this in the back of our mind. When we talk about EPSPs and IPSPs, these are small changes in the membrane charge. These will only happen below threshold. So EPSPs and IPSPs, they only happen below threshold. Why that'll help you is everything that happens below threshold is chemically gated channels that we're using. So I remember when I hit threshold, voltage gated channels take over. These small positive changes or small negative changes happen when I'm below threshold, and they're caused by chemically gated channels. So excitatory postsynaptic potentials, what helps me with that is chemically gated sodium channels. When we're talking about inhibitory postsynaptic potentials, which kinds of chemically gated channels lead to those? Which kinds leads us to inhibitory postsynaptic potentials? Perfect. Yep, we're typing. I'll type with you. If I want my membrane charge to get more negative, there's two ways to do that. The first way to do that is to bring in some chloride, so bring in some negative charges. The second way to do that is to spit out some positive charges. So the two types of channels that help me to generate IPSPs are chemically gated potassium channels and chemically gated chloride channels. The one type of channel that helps me to make an EPSP is a chemically gated sodium channel. Thumbs up, thumbs down. How are we feeling? Or send me some other emoji that better captures your current state. Okay. We're feeling decent, it looks like. I'll take decent. It's just going to take some review, right? So if we're kind of tracking now, that means we'll track even better later. I know that somebody had asked me to review summation, and here's my, I can get it to share. Yep. Here's my summation picture that ties in what we were just talking about. EPSPs and IPSPs. So excitatory changes that make me more positive and inhibitory changes that make me more negative. What we see on this picture, what's happening right here, is the process called summation. And again, think about summation. It's literally math. It's math with those ions. So I open up a chemically gated sodium channel. So I sent a neurotransmitter right here, and it opened up a chemically gated sodium channel. Sodium rushed inside, my membrane charge got positive. And then I got another neurotransmitter right here, and this neurotransmitter right here opened up more sodium channels. More sodium came inside. But then I got another neurotransmitter. I received another one right here. This one that I received opened up a chemically gated chloride channel, for example. Chlorine rushed in, made my membrane charge more negative. Now I've got a little period of time, nobody's talking to me, I'm just listening. Oh, here's a new neurotransmitter. This one opened up a sodium channel again. Here comes that positive sodium, except now I got another negative message. Maybe this one opened up a chemically gated potassium channel, and I'm spitting out all of my positives, getting more negative, until I receive this message right here. And this message opened up chemically gated sodium channels. Again, in rushes my sodium, all of it rushing in, enough of it rushed in to get my membrane charge to negative 55. At negative 55, or at threshold, this is the point where I can start using voltage gated channels. So my voltage gated sodium channel will allow me to get all the way up to positive 30. Once I get up to positive 30, I need to get back down to normal. My voltage gated potassium channels get me back to normal. I go a little too negative, right? But eventually I get back to normal. Remember from our discussion on Wednesday that we had two kinds of summation. Summation, here let's type it as a review for ourselves, just means we're adding messages. Summation means we add messages together. Our goal, again, is to hopefully get to threshold, because that means I get to talk to my neighbor. Two kinds of summation that a neuron can do. We can do what's called temporal summation, and we can do what's called spatial summation. The biggest difference between temporal summation and spatial summation, the best way to think about it, in one of these types of summation, I only get messages from one other neuron. Which of these two types of summation is one neuron talking to me repeatedly? Which one is one neuron talking? Yeah, so in temporal summation, only one, here let's use some of our words from earlier in the lesson, only one presynaptic neuron talks to me. In temporal summation, it's one presynaptic neuron that neuron is sending me lots of messages over a period of time. So remember that temporal meant time. So if the example we gave is if one person sends you 99 text messages to try to communicate something to you, that's temporal summation. One presynaptic neuron talks to you repeatedly over a period of time. That's temporal summation. So when we talk about spatial summation, in spatial summation, that is multiple neurons talking to me, multiple neurons talking to me. Does anyone remember, what does that spatial word mean? Do we remember what spatial meant? Yeah, so spatial just means space, like Nicole said, absolutely. With spatial summation, a neuron is receiving messages over a whole bunch of different spaces on it. So some of my dendrites are receiving positive messages, some of my dendrites are receiving negative messages. Spatial summation is when I take the messages that I receive from a bunch of different neurons that are talking to me at a bunch of different places on my cell body, and I add them together to figure out whether or not I should be talking to my neighbor. So spatial summation, technically spatial summation means space. Different spaces on my neuron are talking to different other neurons, different presynaptic neurons. Temporal summation, it's all about time. One neuron talks to me over a long period of time. For my friend who specifically wanted us to cover summation, help me out with any other questions that you have. Jacqueline asked a good question. Can they both receive EPSPs and IPSPs? So in other words, can I use both of these processes with either, or excuse me, are there EPSPs and IPSPs in either of these? Yes, we could add them together. That way it comes down to if I'm doing temporal summation and I'm getting EPSPs and IPSPs, it means that my original neuron is spitting out different neurotransmitters. So if my presynaptic neuron can spit out different neurotransmitters, that would allow me to have EPSPs or IPSPs. It's more likely, and what we see in this picture here, it's more likely that we're going to have the two kinds of messages if I'm listening to more than one neuron, though. So it's more often going to be spatial summation if we're adding together EPSPs and IPSPs. Generally, a neuron only spits out one kind of message, but if it made more than one neurotransmitter, then sure, we could conceivably have EPSPs and IPSPs in temporal summation as well. Good question. Okay, so we're looking at a phrase from the homework. It says we're trying to figure out so the answer choices are EPSPs and IPSPs, correct, Vanessa? No. So I don't know what the answer choices are then. That would help me. Okay. So we're talking about the types of summation. So here's the question. The question says that we're trying to figure out what occurs when neurotransmitters attach to chemically gated channels on the postsynaptic neuron's membrane. So the answer options are temporal summation, spatial summation, or both. And yes, our friends in the chat are saying correctly that is what happens in both kinds of summation. Neurotransmitters attaching to channels and us adding together what those channels let in, the ions that those channels let in. That's something that I do with both temporal summation and spatial summation. So that, yeah, like I said, the answer would be both. That's correct. Of course. When we do summation and we hit threshold, when we get to this value where my charge is different enough to open up voltage gated channels, there's two things that I want to have happen. I want to have my Axon Hillock depolarize all the way, but I also want my entire axon to depolarize all the way. Because remember, down here at those axon terminals, or down here at those synaptic end bulbs, whatever you want to call them, we got to depolarize down here to get those chemically or those voltage gated calcium channels to open up. So it's all well and good if I hit threshold right here, but I've got to get that hitting threshold all the way down my axon and all the way to my axon terminals for this neuron to be able to talk to its neighbor. And that's where we get into the action potential. Can you help me in the chat? What page do we have this picture on? Where do we talk about action potential? Okay, we're saying page 11. Perfect. Hey, guess what? This picture is literally the exact same picture that we had in the muscles chapter. The way that an action potential works in a muscle cell to get my signal to contract from the neuromuscular junction all the way down into the middle of a muscle cell, that process we use there is exactly the same as the process that we use when a neuron is depolarizing its membrane. So here's how this works. Let's add for ourselves our little Axon Hillock right here. So see my little Axon Hillock? I'll label it. Here at the Axon Hillock, we have, we've added together those messages. Say we had enough EPSPs, enough excitatory messages, that we got to -55. The charge on my membrane right here is -55. Remember that at -55, voltage-gated sodium channels open up. Voltage-gated sodium channels bring sodium into the cell, and they make the inside of the membrane go more positive. Now remember, from when we were talking about muscles, if this part of my membrane goes positive, the voltage-gated channels that live next door say, oh, we're doing this thing. We're going to open up too because we've hit threshold. The voltage-gated sodium channels that live here get opened up because the membrane charge got positive. So these voltage-gated sodium channels then open up themselves. This part of my membrane on the inside gets positive. Well, if this part of my membrane gets positive, the voltage-gated sodium channels that live right here say, oh, we're doing this. These voltage-gated sodium channels open up as well. Now this part of the membrane is positive. And we can predict where this is going because then these guys, the voltage-gated sodium channels that live here, see, oh, we're positive, I'm going to open up as well. This is the process that a neuron uses to get a positive charge from the very beginning of the axon all the way down to those axon terminals. Because if one part of the membrane gets positive, its neighboring part's going to get positive too. And when this part gets positive, its neighboring part's going to get positive. And when we're positive here, we're going to get positive here as well. An action potential is just a fancy word for changing my membrane charge over a distance. It's a moving membrane charge. First we're positive here, then we're positive here, then positive here, all the way down. But remember that when a membrane gets up to positive 30, it realizes, wait a minute, I don't like this. At positive 30, we open up those voltage-gated potassium channels, the ones that I use to repolarize the membrane. They start spitting out potassium to get the end side negative again. And so when this part of the membrane hits positive 30, those voltage-gated potassium channels will open up to get it back to negative. And when this part of the membrane gets to positive 30, we're going to open up the voltage-gated potassium channels here to get this back to normal. So what you see if you watch an action potential travel down a neuron's membrane, first the voltage-gated sodium channels open to make it positive, then the voltage-gated potassium channels open to make it negative again. And next door, first the voltage-gated sodium channels open to make it positive, then the voltage-gated potassium channels open to make it negative again. Both types of channels are used to move my signal from the axon hillock all the way down to where my axon terminals would be here at the end. What questions do we have about the action potential? About the way that a signal moves? Yeah, once again, processing. That's fine. Okay, Nicole asked a good question. She asked about what happens when that charge gets to the end. So let me draw a really funky axon terminal here. She asked, when my action potential gets down to the axon terminal down here, we know that when we hit positive 30 down here, we open up those voltage-gated calcium channels. The question is, would we also open up voltage-gated potassium channels? To help us answer that question, let me ask another question. What's the job of voltage-gated potassium channels? What's the purpose of those voltage-gated potassium channels? Yeah, so voltage-gated potassium channels, their goal is to repolarize the membrane. If I depolarize my membrane down here, I still want to repolarize it. I still want to get it back to normal, because even though this is the place where I spit out neurotransmitters, I don't want to leave this place positive 30 either. So at the same time, when I hit positive 30, not only am I going to open up those voltage-gated calcium channels to get my neurotransmitters spit out, I'm also going to open up those voltage-gated potassium channels. The potassium channels are going to help me to repolarize the membrane. The calcium channels are going to help me to spit out my neurotransmitters. So when we get to that point on the graph, let me draw the graph again. When we get to this point on the graph and we're at the axon terminals down here, two things happen here. First, I'm going to open up my voltage-gated potassium channels to help me go back down. But second, or kind of actually at the same time, I'm also going to open up those voltage-gated calcium channels to help me spit out neurotransmitters. See, once we start to go back down, we start to get negative again, the voltage-gated calcium channels are going to close. I'm not going to spit out neurotransmitters forever. My voltage-gated calcium channels are open here at the top, but as I start repolarizing, they start closing again, and I'll stop releasing neurotransmitters. Yeah, so great question. For most places, At the peak of my graph, I'm just going to spit out potassium. But when we're at the axon terminal, I'm also going to bring in the calcium. Nicole asked if voltage-gated calcium channels are seen on muscles. They are not. Does anyone happen to remember what kind of channels do we have on the sarcoplasmic reticulum of a muscle cell? They were attached to a voltage-sensitive protein. And that voltage-sensitive protein pushed them open. Which kinds get pushed open? Yeah, mechanically gated. So the calcium channels in muscle cells are mechanically gated calcium channels. The calcium channels on neurons are voltage gated. So that's a good clarifying question. Might be a good flashcard for you to make for yourself. The kinds of calcium channels on a neuron are voltage-gated channels. The kind of calcium channels in a muscle fiber are mechanically gated. Yeah, so unfortunately, it's not exactly the same, Nicole. But yeah, both of those types of cells use calcium to make stuff happen. So it's a good tie-in to make. Since we just talked about the action potential here, let's use that. Oh wait, here, I gotta show you guys a joke because this one's pretty good here. I should have showed it earlier, actually. It's our friend Joseph's guy. He says, I don't always reach threshold, but when I do, I fire an action potential. So here's my favorite graph, right? This whole process where we go up and then we go back down, we call this an action potential. So what he's saying is I don't always hit threshold, but if I hit threshold, I'm going to go up and I'm going to come back down. So there's our little humor here to help us through this Friday. We don't always reach threshold, but when we do, it's go big or go time. We're going all the way. I love old-fashioned memes, Hannah. Yeah, I made the Botox one myself, but I found this one online. Somebody else had made this one, so I was pretty happy with that. I do have a couple others too today, so remind me to show you a couple more before the day's done. Back last spring, apparently I had more time to search for memes because I have a few more memes in my my repertoire for these ones. So let's talk. We just mentioned when we were looking at our picture there before the meme, we talked about how messages go along the axon of a neuron. We're using those voltage-gated sodium channels and the voltage-gated potassium channels to get my message from the beginning all the way down to the end. What we're going to talk about now is why it's good for a neuron to have a myelin sheath around its axon when we're thinking about the way that a neuron sends a message from its cell body down to its axon terminals. So remember that when we were in lab talking about myelin, we said that some of the jobs of myelin were to kind of insulate a neuron or there to help it send a message faster. So having myelin, that's my anatomy, means that I send a message faster. That's my physiology. Let's look up close at my ion channels to help me see why that's the case. Can you help me out in the chat? What page do we have these images on? Okay, so we're very close to the end of the packet here. So we're page 16, 17 here, where we're talking about myelin on the neurons. What we're looking at first, this first half of my picture up here, is an unmyelinated neuron. So I'll type that for us. Unmyelinated just means we don't have a myelin sheath on this neuron. Unmyelinated, bear with me, neuron. Here we go. Unmyelinated neurons don't have a myelin sheath. When we depolarize, or when we first start changing the membrane charge on an unmyelinated neuron, so here's where we hit threshold. As I start to try to send that signal along my axon, notice how my signal starts to get a little bit weaker pretty quickly. So I go from having a really strong signal, I can see it big and bold right here, to getting a little bit smaller. When my signal starts to get smaller, I don't want to lose it. I don't want it to disappear. So I'll actually remake that signal using some more voltage-gated channels. So we let in some more sodium here, but then I start to get a weaker signal again. I have to make that signal again. Well, as I start to send it again, it keeps getting weaker, and I have to remake it weaker and remake that signal. In an unmyelinated axon, because there's all this membrane space that's exposed to the environment, my signals get weaker faster. Hey, let's do a tie-in here to our channel types. When we're talking about a type of channel that very slowly moves ions, and we're kind of dripping ions, if you will, what was that type of channel? Yeah, exactly. Okay, so here's my problem with an unmyelinated axon. Covering that unmyelinated axon, the parts right here of the membrane that are exposed to the environment, I've got a ton of leakage channels. And leakage channels, they just kind of drip slowly. But if I'm constantly dripping slowly, I end up losing the strength of the message or the signal that I'm sending. So to make sure that this doesn't go to a complete point where it's zero and there's no message left, I can only travel a little distance before I have to remake that message. And so then I can start to send it again, but those leakage channels are weakening it until I remake that message and send it again. In an unmyelinated axon, I'm going to have leakage channels giving me trouble, eating up my signal, essentially. When we look down here at a myelinated neuron, a neuron that does have a myelin sheath, here's where I made my signal at the axon hillock. But because I have a myelin sheath that covers up those leakage channels, my signal will stay stronger a little bit longer. That means that I can send it faster in this place before I have to open up channels again. And I'll open up those channels and rush in my sodium again, but I can keep going for a longer distance before I have to rush sodium in again. So my myelin sheath blocks those leakage channels, which means my signal is going to stay stronger for a longer period of time. Plus, while we're at it, if I only have a little small space where ions would be able to rush into my axon, my ions tend to congregate here. So remember, normally inside the membrane of a cell, it's -70. That negative that's normally inside the cell means all of my sodiums that are chilling out here with their positive charge, they're attracted, because opposites attract, to this part of my neuron, the one part where they can see, if you will, or feel the negative charge that's on the inside. So my sodiums chill out right here. They're just waiting for the voltage-gated sodium channels to open. When that charge comes along here and opens up these channels, there is all kinds of sodium waiting to rush inside quickly. And that sodium rushes inside, which makes the inside of the axon positive, and that it stays positive enough until we reach the next place. Hey, remind me, this was a lab term. What's the name, the technical name for this space right here where there's no myelin? What's that space where there's no myelin called? Yeah, exactly. The space with no myelin are those nodes of Ranvier. Nodes of Ranvier. The little tiny spaces where there's no myelin on the membrane of a myelin. I know Jacqueline had fun with that one, nodes of Ranvier. This is the one space where there's not myelin on the membrane. It's the only place that I have to remake my signal. So my signal goes a lot faster. The name of how I send a message down a myelinated axon, this is something called saltatory conduction. Saltatory conduction. Or sometimes you'll see it paraphrased jumping conduction. Jumping. I spelled that wrong. When I am sending a message down a myelinated axon, I build it right here, and then I get to jump to the next gap to build it again. And then I get to jump to the next gap to build it again. I'm just jumping my way down the axon, as opposed to an unmyelinated axon, where really quickly I have to go through and rebuild this signal all the time. An analogy that I used with my students in the spring was thinking about if you are in a hallway and we have spaces where every 3 feet there's a door, and you have to open up that door every 3 feet. For you to be able to walk through this hallway, we've just got to keep opening doors every 3 feet. As opposed to when we're talking about a myelinated axon, maybe it's every 12 feet or every 15 feet I open a door. And then I get to go another distance, another 15 feet, and then I open another door. If we're trying to do a race and trying to get through as fast as possible down this hallway, if I have to stop and open doors every 3 feet, I'm going to go a lot more slowly than if I get to go 15 feet and then open a door, another 15 feet and open a door. So maybe consider that when you're thinking about the speed that messages can travel. If I constantly have to reopen voltage-gated channels, which is what I'm doing with an unmyelinated neuron, if we're constantly reopening those channels, it's going to take a lot longer for me to get my message all the way down compared to just opening it every so often, compared to just using the nodes of Ranvier. Yeah, so Vanessa asked about the process called continuous conduction, and that is what we do in an unmyelinated neuron. So this process here, where every 3 feet we have to open up a door, or pretty much every part of the membrane we have to do this process on, that's called continuous conduction, because I'm continuously opening up my voltage-gated channels to build an action potential. So Jacqueline asked, but we don't see that. So there are neurons in the body that are unmyelinated. And with unmyelinated neurons, we would see the process called continuous conduction. The vast majority of neurons in your body are myelinated neurons. So for the most part, the way that you send messages is using saltatory conduction. There are some places though, for example, in your brain, where we have a lot of unmyelinated neurons. Those unmyelinated neurons are going to do continuous conduction. So it's going to take them a little bit longer to send a message because they have to depolarize every part of this membrane, not just the nodes of Ranvier, because they have no myelin everywhere. So the direct answer to your question, Jacqueline, we do see continuous conduction with any of our unmyelinated neurons, but when we have myelin, we do not do continuous conduction. We do saltatory conduction instead. Does this process, or what makes these two processes different from each other, does that make more sense? Oh, definitely, Jacqueline. Yeah, we're going to talk about the refractory period next. Sure. Yeah, Nicole's got a question. Go for it.
It's a question from the homework, and I just want to make sure that I understand the anatomy of the neuron, right?
Okay.
Right here, I'm pointing to this is axon, and then these are the terminal. What is this part right here? Because there's no myelin sheath on it, but I wasn't sure what to call that.
Yeah, so I would probably consider that to be part of the axon terminal, because it's not still the axon anymore. So kind of once we've branched, we're close to that axon terminal. Technically speaking, I know earlier today you asked about the synaptic end bulb. Technically, at the very end of these is the only place where we're going to spit out neurotransmitters, is at those synaptic end bulbs or the actual axon terminals. We're not going to get super specific to what I might call this region here. If I'm asking you questions on the homework assignment, focus on the longer part that on this one is definitely myelinated compared to this one up here that's definitely not myelinated. Because it's anatomy, I bet they have a special name for this part. I'm going to be honest, I don't know the special name for it. For the sake of our class, we're going to consider all of this the axon terminal. So the axon ends right here where we split into all of these branches. That's what we'll consider it. Yeah. Okay, so the one other thing I want to make sure that I hit with you guys is the refractory period. So here is, what page is refractory period on? Can you guys help me out with that? Okay, so it starts on page 14, bleeds onto 15. Perfect. So when we talk about, let's type its name up here, the refractory period of a neuron, can you help me out in the chat? What's going on in the refractory period of a neuron? Why do we even care about the refractory period? Does anybody know yet? I may not have gotten this far. Yeah, so Tierney's right. She's saying that we can't have, nothing happens, basically. In the refractory period of a neuron, technically speaking, no new action potentials can be made. That's a fancy way for saying that a neuron can't receive any new messages. Here's the simplest way or the easiest way to think about the refractory period. During the refractory period, I am responding to one message. I can't start a response to a new message at the same time. I'm overwhelmed entirely with responding to one message. I can't respond to another. The reason that we have a refractory period in our neurons is because of our voltage-gated sodium channels. Our voltage-gated sodium channels. This, by the way, is an underline highlight star idea. The only reason we have a refractory period, or the type of channel that causes the refractory period, are voltage-gated sodium channels. Voltage-gated sodium channels. Can you remind me in the chat, what's their job? What do they do when they're activated? Yeah, they depolarize the membrane, they build us an action potential, absolutely. So to put it in the simplest words possible, voltage-gated sodium channels make the membrane more positive. Biggest picture, maybe what we could say a voltage-gated sodium channel does, is it makes the membrane more positive. So that's the part of my graph over here where we're getting more positive, right? That's the job of a voltage-gated sodium channel. Now, we see a little flow chart here that shows me the three forms or the three shapes that a voltage-gated sodium channel can be in. So when I'm at resting membrane potential, I'm waiting to hear a message, notice that my voltage-gated sodium channel, the channel for my protein or my ions to go through is closed. There's not space in here. My gate has closed on it. When I am depolarizing the membrane, when I've received a message or when the voltage on my membrane has gotten to threshold, my channel changes shape and there's space for sodium to rush inside. Now, all through the process of depolarization, all through as we're going up here and my membrane charge is shooting up higher, all through this process, I have my voltage-gated sodium channels open, allowing sodium to get inside. When I get all the way up to positive 30, I don't want any more sodium to come inside. I'm up to positive 30. We are way too positive. I want no more sodium rushing in. When I get to positive 30, notice this little stopper right here at the bottom of my picture. Think about this kind of like a stopper that you'd put in a bathtub. So we're blocking the drain in the bathtub. When I put in this little stopper right here in my voltage-gated sodium channel, sodium might try to get back inside, but we've got a plug. Literally, we can't get any more sodium inside the cell because of this voltage-sensitive protein down here, this part of my channel. It chills out. It's totally cool when we're in the process of depolarization. When we get to positive 30, this protein says, wait a minute, we are way too positive. We don't need any more sodium. At that point, notice here how we've got the word inactivated next to this protein. When I get to positive 30, this channel is completely inactivated. There's going to be no more sodium moving. Nicole asked which side the plug is on. The plug is on the inside. So when the inside of my cell gets to positive 30, I put this plug in place to stop my sodium channels from bringing in any more sodium for me. Now, over time, when I've plugged up my sodium channel and no more sodium comes in, and I've opened up my potassium channel to let potassium go out, my membrane charge starts to get more negative. And it gets more negative and more negative. It's working its way back down. By the time I get back to around the threshold value on my neuron, I've gone from this plug being in place holding closed my channel to my channel closing itself because I'm below threshold again. When this channel closes itself, that inactivation protein goes back to being normal because my channel's not open anymore. So let's retalk this process. Step 1, I'm waiting to hear a signal. My voltage-gated sodium channel is closed. Step 2, I have hit threshold. I open up my voltage-gated sodium channel. Sodium rushes inside. Step 3, way too much sodium inside. My inactivation protein slides into place, blocks this channel, and now my voltage-gated potassium channels can do their job to get me back down to normal. Once I get closer to normal, I will change back to my normal shape where I'm closed and I'm waiting to receive a message. The reason we have the refractory period where a neuron can't receive any new messages is because the point in time, or excuse me, when a neuron receives a message, what it does is it opens up these channels. These are the channels that open when we hit threshold because we received a message. These are channels that are opening up to allow us to respond to that message. During what's called the absolute refractory period, where I can receive absolutely no more messages, my voltage-gated sodium channels are already open. I can't receive any new messages in the absolute refractory period because my voltage-gated sodium channels are busy responding to the last message. They're open and letting sodium inside, or they're open and I've got that plug inside of them. The way that I respond to a message is to open these channels up. If they're already open, I can't open them again. They're still already open. So in this absolute refractory period, the channel in my voltage-gated sodium channels is open. I can't do anything about that. I can't respond. We also have a place that's called the relative refractory period. In the relative refractory period, it's not that I can't receive any messages, it's that you have to kind of be yelling for me to hear you. We've got to have a strong message to make me respond. The relative refractory period, once we get back to threshold, my voltage-gated sodium channel is in the correct shape. But what's giving me trouble is the fact that my voltage-gated potassium channels are open. These voltage-gated potassium channels are dropping my membrane charge really fast. So in this relative refractory period, it's kind of like if you go skydiving and you're shooting down, but while we're in this relative refractory period, if we pull up our parachute, maybe we can start going back up. But we're skydiving here. My membrane charge is dropping down really, really fast. You're going to have to work hard to get me back to threshold so that I can respond to another message. So in the relative refractory period, if my message is loud enough, in other words, if I open up enough chemically gated sodium channels to get back to threshold, I can respond to a message. I've got to get to threshold though, and I'm actively making my membrane charge more negative. It's going to take a lot to make me go more positive. During the absolute refractory period, I don't care how many chemically gated sodium channels you open up. My voltage gated sodium channels are out of commission. Either they're already open and pumping in sodium, or they're open and they're blocked. It doesn't matter. I can't respond at all. I can't do anything in my absolute refractory period. So Gloria asked, which one is hyperpolarized? So you're asking what shape the sodium channel would be when we're hyperpolarized? Is that the question, I think? If I understand it right? When a neuron is in this stage right here, when it's hyperpolarized, my voltage-gated sodium channels would be here. So I have, so here, let's add a note here. This closed state and normal state, this is what I'm at from resting membrane potential until threshold. Resting membrane potential until threshold. When I hit threshold, I change to this shape where I'm open and sodium comes inside. When I hit positive 30, so when we get to positive 30, what happens is that inactivation gate protein comes in and blocks the channel. This protein is inactivated. As I start to repolarize my membrane, as I go from positive 30 back down to when I get back to threshold, so when back to negative 55, once I get back to negative 55, I've closed up my channel and I spit out that plug. So when I get back to threshold, I'm back to here. And now I'm ready to listen for a new message. So when I am below threshold, I'm closed. I hit threshold, I open up. When I pump in so much sodium that I get it to positive 30, I put in the plug. And now I just wait and wait and wait until my membrane charge drops all the way back to the threshold value. At that point, this channel is going to close and my inactivation protein moves out of the way because it knows that no more sodium is going to sneak inside. So when we get back to threshold and the channel closes, my inactivation protein moves out of the way from there. Let me read Nicole's question. Yeah, so the question, if I am interpreting it correctly, is basically when does the relative refractory period begin? The relative refractory period begins at -55 when I'm on my way back down. So the absolute refractory period starts the moment that I open up my voltage-gated sodium channels. My voltage-gated sodium channels are completely open all along this way, I get up to here. This is when I put in that bath plug, when I'm stopping the flow of sodium. I'm still in the absolute refractory period though, because my voltage-gated sodium channel is technically still open. It's just plugged. Nothing's coming through. So I work my way back down. I'm getting more and more negative until I get back to my threshold value. When I get back to my threshold value, the channel is closed again. When the channel closes again, when it would be possible for me to now go through and open it again, that's when I hit the relative refractory period. When it's possible for me to reopen that channel for a second time, that's when we hit the relative refractory period. So until I get back to threshold, that channel is still open from the first message. I can't receive another message. Once I'm back here to threshold and I've closed that channel again, now it would be possible for me to get another message. Hard, but possible. That's why I'm in the relative refractory period. What other questions do we have? Clarify what you mean by message, Cole. For my friends watching the recording, we're asking if the message is for voltage-gated sodium or voltage-gated potassium channels. What I want to clarify here is the only kind of channel that actually causes a refractory period, it really is all about the sodium channels. My sodium channels, my voltage-gated sodium channels, are the channels that I need to open to respond to a message. My potassium channels don't help me respond to messages. First, a sodium channel has to open up. So when I reach threshold, or when I've received the stimulus, my Axon Hillock has made me more positive, I open up a voltage-gated sodium channel to help me send the action potential farther. If I'm trying to send another action potential, if I'm trying to depolarize the membrane again, the only kind of channel that I can use to depolarize the membrane are these voltage-gated sodium channels. If they're already open from the last message I received, I can't receive a new message. They're already responding to the first message. So for as long as my voltage-gated sodium channels are open in some capacity, so as long as they're either open and letting sodium inside, or they're open with a plug, as long as my voltage-gated sodium channels are open, I absolutely cannot respond to another message. Once I get them back to the correct shape where they could open and they could let new sodium in. Now I've entered what's called the relative refractory period. The relative refractory period is when I could receive a message. It's just going to be hard. The reason it's going to be hard is because my membrane charge is dropping really, really fast, or it's too negative compared to what it wants to be. So it's not that there's anything wrong with my potassium channels. They're doing their normal job. Their normal job just makes it harder for those sodium channels to open up again. So once I am at threshold and below threshold here in the process of repolarizing, I'm in that relative refractory period. If I were to bring in enough sodium, that might help me bounce back up to threshold and reopen these guys. But these are what have to open for me to be able to respond to a message. If they can't open, I'm not going to be able to respond. Okay, Gloria, yeah, so when you ask what hyperpolarization means, yes, that means that we're more negative. So there's two kinds of messages that neurons send to each other. They send depolarizing messages. That makes the membrane more positive. And they send hyperpolarizing messages. Those make the membrane more negative. So yeah, so the point on my graph that we are hyperpolarized when we're below normal, the point where we're hyperpolarized, we are in the relative refractory period. The reason why this makes us be in the relative refractory period is because anytime I'm when I'm down here, anytime I'm lower than normal, that's going to make it harder for me to get up to threshold. I've got to get up to threshold to be able to respond to a message. So in the relative refractory period, you just got to give me extra sodium to make me get up to threshold. I can get there because my channels are closed and they're waiting to reopen. So when I'm extra negative, or when I'm in the process of getting more negative, that's going to make it harder for me to respond to a message. So that's my relative refractory period down here. Let me check what Christina said. Hyperpolarization is when the membrane charge is getting more negative. Yep, and so when I'm out here in the more negative time, I can use those leakage channels, yeah, to leak me back to normal resting potential. Yeah, so over time, I'll use those leakage channels and remember that sodium potassium pump to get me back up to normal. Once I get back to normal, once I'm at resting membrane potential, then we're back to normal and we're not in the refractory period anymore. So the refractory period is the point in time where either I can't hear any messages at all, you can't talk to me that you can make me do a new message, or if we're in the relative part of the refractory period, you're going to have to scream at me to make me respond to your message. It's hard for me to respond. It's possible, but it's hard. So let me see, Christina, I'll get you in a moment here, Andrea. Christina says the difference between the absolute and relative refractory periods would be because the absolute refractory period cannot generate a new action potential, and in the relative refractory period, we would be able to if it was strong enough. Yes, absolutely. So absolute refractory period, no messages, zero, can't do it. Relative refractory period, if we try hard enough, we can. Oh, no worries, Andrea. You're good. Keep the questions coming. What else are we wondering about? Shoot me an emoji. How are we feeling about the refractory period? Yeah, Nicole's not loving it. Awful. I'm sorry, Gloria. Yeah, it's going to take some time. Here, let's do a meme to try to make it better. So we're going Lord of the Rings on you now. He's saying one does not simply conduct an action potential. So depolarize the membrane, all that jazz, right? When we're in the absolute refractory period. Lord of the Rings fans, or not as many Lord of the Rings fans in the audience? Christina is. Okay, got a couple people. Oh, Jacqueline's never seen it. Oh man. So we simply cannot do it. If we are in that absolute refractory period, no new messages. Cannot send more messages. If we're in the relative refractory period, you just got to scream at me. We got to be loud. So when we're right here, when my sodium channels are the wrong shape entirely, we're in the absolute refractory period. Can't hear any new messages. Once my sodium channels get back to the right shape, I could hear a message, but it's going to be hard because I am getting more negative or I'm extra negative compared to what I should be. The refractory periods, when it's harder or when it's impossible for a neuron to respond to another message. That's the big idea with the refractory period. That is all I've got for today. What parting questions do we have today? Here, I'll draw you a penguin too. I can't remember if I did a penguin last time. Yeah, so I bumped into the SI session today, unfortunately. I apologize about that. I did not consider that when I switched to Friday for today. She was starting her session today at 11, so please head on over that way if you're interested in another take on this material. Head on over to that session now. I will get our schedule for next week's sessions posted, hopefully either today or sometime tomorrow. It's going to be Monday, Wednesday, Thursday, again, as usual. And we will be talking about the brain and the lab stuff. So don't forget, it's Friday today, which means that we've got some visible body assignments that are due today. And then we've got the lab homework assignment and the lecture assignment that are both due on Sunday. So keep those in mind. That's all I got for today. Have a great weekend.